Hi science friends, let's go ahead and get started on the second part of the cellular respiration segment. So let's review just a little bit from yesterday. What is the function of NAD+? You should be able to quickly say that this is an electron carrier. It picks up electrons in cellular respiration and takes them to the electron transport chain. When it picks up electrons, it becomes NADH, okay? And as it gains electrons, it is reduced to form NADH. So for those of you that watched the video yesterday, you will have seen that it was an ed puzzle, and so there were a few questions at the end. Some of you all have already watched that. Some of you haven't watched that. It's just a participation grade for completing that. But there, for those of you that have already done it, it's 1:22 a.m. quite frankly on Wednesday night to Thursday morning. So those of you who've done it before now, then some of the answers were marked incorrectly, and so I've gone back and fixed that. So some of them I accidentally hit 2 answers that were correct, and that wasn't the case. So if you were a little confused on that, or if you thought you missed some that you didn't miss, then you were probably right. I messed up a little bit there, so I apologize for that. So let's look at the next part. Explain the terms oxidation and reduction. So oxidation, Leo, is loss equals oxidation. Reduction is gain, and we're talking about loss and gain of electrons. Leo says GER or oil rig, okay? And then next, what's the difference between aerobic and anaerobic respiration? So anaerobic is without oxygen, aerobic means with oxygen, okay? So cellular respiration can occur both ways. Anaerobic respiration is also called fermentation, and we will talk just a little bit about that later on. We'll touch on it today, but talk a lot more about it later on. So let's talk today about the stages of respiration. So for the most part here, we are talking about aerobic cellular respiration. So there are 4 stages in aerobic cellular respiration. Although this first stage, glycolysis, is also the first step in anaerobic respiration. So this is for aerobic or anaerobic respiration. And then these next 3 stages are aerobic only. So glycolysis happens first either way. And then if oxygen is available, then it will proceed to these next 3 steps, okay? If oxygen is not available, then it will opt to move to anaerobic respiration or fermentation. So is about all that we're going to touch on today with anaerobic respiration. So glycolysis happens first. Now, I don't think I explicitly said this in yesterday's video, but it was one of the questions and the end of at the assessment on the ed puzzle. And so cellular respiration happens in the mitochondria. Plant and animal cells both have mitochondria. Plant and animal cells both do cellular respiration. Only plant cells do photosynthesis, but plant cells also do cellular respiration, okay? So please understand that. Now, cellular respiration also happens in both prokaryotic and eukaryotic cells, okay? Eukaryotic cells, meaning they have a nucleus, those are the ones that have all the organelles. Prokaryotic cells are like our bacteria that don't have very many organelles, and so they don't have a mitochondria, but they still do cellular respiration because they still acquire energy and they still need ATP. And so they make that ATP through respiration. But because they don't have a mitochondria, these steps that occur in the mitochondria In a prokaryotic, they occur in the mitochondria in a eukaryotic cell. In a prokaryote, they happen in the cytoplasm. Okay? So the first step of respiration, glycolysis, it happens in the cytoplasm for everyone. Okay? Prokaryote or eukaryote. In a eukaryote, the remaining 3 steps happen in the mitochondria. In a prokaryote, those steps will occur in the cytoplasm. Now, this last step, step number 4, it says it happens in the outer membrane of the mitochondria. So remember, the mitochondria is a double membrane structure, as is the chloroplast, a double membrane structure. So it happens in the outer membrane of the mitochondria. In prokaryotes, it happens in the cell membrane. Okay? So, which borders the cytoplasm there. So, that's very important for the distinction of where these occur in the prokaryote versus the eukaryote. Okay? So, the first stage of cellular respiration is glycolysis. All right? With or without oxygen in the cytoplasm. The second stage is called pyruvate oxidation. Sometimes, on one of your worksheets that you're going to do, this is known as the link reaction because it's kind of just a stepping stone between glycolysis and the citric acid cycle. It's really a small piece of the whole puzzle. So in that, I don't ever personally call it the link reaction, but that is how your worksheet describes it. So just know that it's talking about pyruvate oxidation. The third step is the citric acid cycle, also known as the Krebs cycle. Okay, so it's important that you recognize that by both names. I cannot promise which one AP will use. Both names are very common for that step. And then the last one is kind of known by 3 names. So it's sometimes called oxidative phosphorylation. It's sometimes called the electron transport chain. Most commonly, that's what I will refer to it as. But it's also known as chemiosmosis. Okay? And so probably the electron transport chain is really the best known name of it, but it is where oxidative phosphorylation occurs, and we'll talk about that in a minute. And it's also where chemiosmosis occurs, and we'll talk about that later on as well. Okay? So, let's take a look here first. This is a very important diagram because it overviews, this is a very simple overview of cellular respiration. Notice we have the 4 steps, 1, 2, 3, and 4, glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, the electron transport chain, and chemiosmosis. Okay? Notice also that we're releasing energy in the form of ATP, which makes this an exergonic reaction. Okay? You also see these molecules that you learned about yesterday, NADH, which carry electrons. Okay? And they are carrying them. You see the arrow going here. You see the arrow going here. They are carrying them to the electron transport chain. Okay? You see that this first phase, as we talk about, is happening in the glycolysis is happening in the cytoplasm. And then the remaining 3 phases, this is a eukaryotic cell, so they're happening within the mitochondria. Okay? You may have also noticed that this ATP looks bigger because this third step, as we mentioned yesterday, this 4th step, I don't know why I wrote 2 3s, 4. This 4th step, as we mentioned yesterday, the electron transport chain, is where the majority of the ATP comes from. So this explosion here looks a little bit bigger than the one here or the one here, okay? Because most of the ATP comes in that phase. So, again, This last one is oxidative phosphorylation, as we called it in its name. Okay? These are a different type of phosphorylation, and that's substrate level phosphorylation. And again, we'll talk about what that means in just a minute. But first, I want you to pull out one of your white sheets of paper that I put in the back of the packet that you have. It's just a computer paper, so if you haven't got your packet yet and you have a piece of copy paper, then pull that out. If not, just do it on a piece of notebook paper. But I want to first today talk about glycolysis. So this is step 1, glycolysis. And ultimately what glycolysis is, you should recognize that word lysis. Lysis is a breakdown, and we are breaking down glucose, okay? So in this step, glucose is broken down. It happens in the cytoplasm, and it happens aerobically when oxygen is present, or anaerobically if oxygen is not present, okay? Now, glycolysis, you should remember from yesterday, the chemical formula is C6H12O6. So if it's a 6-carbon structure, right? It's got 6 carbons. So if you break down glucose in half, you're going to get 2 3-carbon molecules. And those 2 3-carbon molecules are called pyruvate. Okay? When glucose is broken in half, it forms 2 3-carbon molecules called pyruvate. Each of them 2 pyruvates. Okay? Now, when we break down glucose, we talked about yesterday that you break a bond, you release energy. So in this process, we're releasing energy in the form of ATP, okay? And when you break a bond, you also release the electrons that are held in that bond. So we've got 2 electrons that are going to be picked up by our electron carrier, NAD+, and once it picks them up, it's going to be formed into NADH, which is going to take them to the electron transport chain. Okay? And that's step 1. That's what happens in glycolysis. Glucose is broken down into 2 molecules of pyruvate. ATP is released as an energy for the body, or the cell rather. Electrons are also released in the breakdown because if glucose is losing electrons, so loss of electrons is oxidation, all right? And so it's been oxidized, so we see these series of oxidation and reduction reactions. NAD picks those up to form NADH, which takes them to the electron transport chain. Okay? And that's step 1. Now, in step 2 is called pyruvate oxidation. So pyruvate is going to be oxidized. So that means it's going to lose electrons. Okay, and so our pyruvate came from here. It's the same pyruvate. And so when it gets oxidized, the molecule of pyruvate, I don't know what it looks like, honestly. I couldn't draw it. It doesn't matter. But it's got an oxygen on it. Okay, and so essentially what this molecule looks like, it's a structure. We'll just make something up. It's a structure, whatever, it's big. I could find a picture of it and show you, and I will at some point later in the PowerPoint. It's on down the road. But it's broken down. What happens, though, is an oxygen is broken off of it, okay? And when that bond is broken, guess what's released? 2 electrons, and so it's oxidized because it's lost electrons. Oil rig. Oxidation is loss of electrons, okay? And so this remaining molecule that doesn't have the oxygen on it is called acetyl-CoA. And that stands for coenzyme A. We talked about coenzymes a little bit briefly. Acetyl-CoA. All right? So pyruvate is the whole thing here, this whole picture. When the oxygen gets taken off, it forms acetyl-CoA. All right? And it's lost those electrons. It's oxidized in the process. Okay? That product, acetyl-CoA, then enters the third step, which is the Krebs or citric acid cycle. Okay? That's what you have to get the citric acid cycle going, is acetyl-CoA, which came from pyruvate, which came from our glucose molecule. I can't draw here. From our glucose molecule that we started with, that we ate, that we got in our diet, right? When you eat any type of carb or sugar or anything, all right? So that's what we're talking about here with cellular respiration. We release some ATP along the way. Whoa, I got on my eraser, sorry. We release some ATP along the way, and we release some electrons that got picked up. These electrons also will get picked up and shuttled to the electron transport chain. Okay? So let's go back to our PowerPoint. And here you see this happening. Glucose is converted to pyruvate. Pyruvate is oxidized to acetyl-CoA, and acetyl-CoA enters the citric acid cycle. And then we've got electrons that are moving to the electron transport chain from these processes. Now, let's talk about this substrate level phosphorylation and oxidative phosphorylation and what that means. So the difference in these two is where does the phosphate, PO4, you should remember from that functional group, come from. Okay? And that's what makes it oxidative or substrate level. So in oxidative phosphorylation, the phosphate comes from inorganic phosphate that's just available in the cell. Okay? So you will see this in diagrams written as PI because it's inorganic phosphate. That's what the I stands for, inorganic. And so it's just phosphates that are available in the cell. Those ions are available in the cell. And they get added to ADP to make ATP. And so we see this in the stages where ATP is made. So ATP is made, the phosphates coming from inorganic phosphate in substrate level phosphorylation. In oxidative phosphorylation. I said that backwards. In oxidative phosphorylation. So in this one, okay, inorganic phosphate in this one. In these, in substrate level phosphate, the phosphate is coming from a substrate. So what's a substrate? Well, a substrate's just a molecule in the cell. Now, we talked about substrates with enzymes, right? Substrates are specific to enzymes. And so it's catalyzed by an enzyme here. But a substrate is just an organic molecule that's an intermediate that's available in the cell. But it works with a specific enzyme. Remember that induced fit, that they have to fit well together. And the phosphate comes from that. So it's plucked off of another molecule. So you have a substrate molecule that has a phosphate group attached to it. And ADP steals that phosphate, takes it away from the substrate, and uses it to form ATP. Okay? And so we see that happen here in glycolysis and in the citric acid cycle. Okay, you don't see that happen in pyruvate oxidation. There's no ATP formed there. That's why I said it's kind of a minor stepping stone. That's why in the worksheet it's called the link reaction, because it just takes us from glycolysis to the citric acid cycle, both of which make ATP. So that one doesn't. It's a minor reaction. But the difference in oxidative phosphorylation and substrate level phosphorylation is where the phosphate comes from. Oxidative phosphorylation happens in the electron transport chain and in chemiosmosis. Substrate level phosphorylation happens in glycolysis and the citric acid cycle. Okay? So sometimes cellular respiration is viewed as, specifically as glycolysis, as an investment. So in order to break down glucose, we have to put energy in, okay? So I want you to think of it like you're eating. Like we eat to get energy, right? But it takes energy to eat. It actually takes energy to eat. You have to have energy to chew. You have to have energy for your stomach to digest things. And so you invest energy because you know the payoff is going to be bigger. And the same is true for glycolysis. It takes 2 ATPs to get glycolysis started, to break down that glucose molecule. But ultimately, the process of glycolysis forms 2 ATPs. So glycolysis nets 2 ATPs. There's a net production of 2 ATPs in glycolysis. That number is important, okay? Glycolysis makes 2 ATPs, all right? The rest of the energy is made over here, but glycolysis makes 2, okay? So you may think, Miss Brown, this is a lot. That diagram's a lot. These are a lot of words. We've got glycolysis, we've got glucose, we've got Krebs and citric acid and pyruvate and acetyl-CoA and oxidative phosphorylation and substrate level and all this stuff. And I know it seems like a lot, and it is, but this is really the whole picture. This is really glycolysis, okay? So let's walk through again the parts that we talked about it. We start with glucose, okay? And also it's a 6-carbon sugar, so you see it drawn as a hexagon here because each of those points represent a carbon, okay? And it has to take energy. We have to put in energy, use ATP to break it down. We're going to use more ATP here, so it's got an investment of 2 ATP, okay? But down here, it's generating 2 ATP, and then it's generating 2 more ATP. So 4 have been generated, so it's net 2, okay? Net 2 ATP. All right, so just like if you went to Keeneland and you bet $20 on a horse and you won $40 on the race, you really only netted $20 because 20 of it was what you paid in, right? And so the same is true here. So we've taken our glucose molecule and we convert it to pyruvate, all right? And we've done this energy transfer in the process, okay? Excuse me. Notice that our pyruvate is a 3-carbon molecule. And it makes 2 of them. This is only showing 1 of them, but it makes 2 of them, okay? And so that's important. We see here that electrons are released in this breakdown, and the NAD picks up those electrons and becomes NADH. We also see here substrate level phosphorylation. So to make this ATP here, we have to get those phosphates from somewhere to add to ADP. So where do those phosphates come from? Well, they come from a substrate. So here is the substrate that's being used, 1,3-bisphosphoglycerate. I don't even know what that is. I don't care what it is. But notice that it has 2 phosphates on it, okay? But in this diagram, after it's been reduced, because it lost electrons, it only has 1 phosphate. The overall structure looks the same. This part of the structure is the same as this part, right? But what it lost was this phosphate. And there's 2 of these molecules. And there's 2 of those pyruvates. There's where you see the 2 indicate there's 2 of them. And so this phosphate was plucked off of that substrate and added to ADP to form our ATP. Okay? So while I want you to get the big picture and understand the parts, realize that there's a whole lot of steps that we're not touching on. Okay? So hopefully this diagram is helpful to you. Students always seem to really appreciate when we draw these diagrams because it helps them to kind of break it down and see it in their head and see it and write it in a way that's not just in your notes packet. So we will add onto our diagram more tomorrow and draw new diagrams and continue to grow as we grow our knowledge in cellular respiration. So if you all have any questions, let me know. And if not, then you all have a great day.